without delaying, now I'll welcome all the attendees, sirs and madam, in this Science Dialogue series. Most of you are aware of about this series. Those who are not aware, so this is a free Science Dialogue series. Purpose is to disseminate knowledge in biological science, bioscience stream. And always we invite the speakers from renowned institutions, or if he or she has very good reputation and very good experience, so we call them. We don't call randomly, we call those who are from good institutions. In this series, today's speaker is Dr. Ritu Pasrija. She is my senior also, 2-3 years senior. I was also pursuing PhD, then she left after 2-3 years. And so she is, as you know from the flyer, she is working at Haryana as associate professor. But before joining there, she has worked in company also, run vaccine. As you all know, that time it was very renowned. उस टाइम में मैडम का डायरेक्टली पीएचडी के बाद वहां पर हुआ था, है ना, रन वैक्सी में. So it was very good company. And madam did her PhD from very good reputed lab in School of Science, School of Life Science, Jawaharlal Nehru University. So throughout you can see she did her PhD under Rajendra Prasad sir, very dynamic teacher, as I know. He has still he is devoting his time for upliftment of science, bioscience. He is working. So she did her PhD from JNU, then joined in Ranbaxy, and nowadays he is associate professor at Haryana. Anna. So with this brief introduction, I welcome madam in the Science Dialogue series, and we are grateful to Ritu madam for accepting our invitation. And I'm sure that you all will learn this new topic. You will not know about all these topics, and I request you all to attend complete lecture. Then only you will learn, and you can interact. No one is in mute mode. You can anytime you can unmute and ask question if you find difficulties. But generally, I suggest you to write in chat box. I will speak and then madam will reply. But if you think it is necessary, you can unmute also and speak and ask question. You will also get e-certificate if you will attend complete lecture and fill the e-feedback form. I will share during this lecture, not before or not after. So you must attend. I can randomly add that Google form and it will close just after this lecture. So you all know this rule. I am just repeating. Most of you are old. If you are free, you can also join our social sites if you don't want to miss any lecture in future. And you can join Google, YouTube to refer older lectures which we have arranged under this platform. Madam, again, you are welcome in this Science Dialogue series. Thank you very much. Now platform is yours. Unmute. Yes, madam.
Thank you. So firstly, I'd like to express my gratitude to Dr. Gopal for inviting me to speak on this platform and allowing me to share my work. Along with that, I would like to definitely mention that I appreciate the efforts that you are taking for sharing the ideas on science so that people learn how we work on any problem, how we design the experiments, how then we complete a story. So I'm just presuming that there is heterogeneous group, and most of them must be students. So keeping that in mind, I have basically tried to make a story of a piece of work that we did in our lab, and I'll just share my slides, Gopal, and just let me know if it is working.
So show entire screen, not the window.
Okay. So for that, I need to put my slides on the full screen mode first.
No, still you have not shared. So see here, there is arrow. So you screen share, है ना, वहां पर जाएंगे. Then you have option कि Chrome tab, window, entire screen. So click on entire screen. Then आपको एक window दिखेगा, उस पर फिर कर्सर ले जाएंगे. Then share का option आएगा. Okay. I thought कि आपने किया है इसको.
I did it during Corona time. We used to take classes, but it's been long now. I'm
So
just
there is
trying to
arrow.
again.
Ha 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 ha.
So,
Yes.
even our lab meetings also happen on this on
Yes.
weekend
So now
days.
whatever you will open, it will be visible. Entire screen किया ना आपने? Yes.
Yeah, I think I did.
Yes.
Yes.
Now full slide mode. Click on that full slide mode. Yes.
Okay.
Yes, it's visible. Well visible. Just hide there. तो जो ग्रीन लाइन है
Okay.
ना स्टॉप प्रेजेंट वहां हाइड कर दी. यस. यस मैडम, इट्स विजिबल. वेरी गुड.
But I can't see you, so I have
Yes,
to.
yes, no need to see. So
Okay.
you just present, and
Okay.
whatever they, if they need something, so they will ask.
Okay.
And
Okay.
after presentation, question answer.
Okay. Okay.
Yes.
So, good evening, everyone, and warm welcome on this evening, Saturday evening. You have spared time for learning some science. So, let's discuss something what we are doing in our lab. So, I'm basically a microbiologist. My training was on fungal pathogens. But apart from fungi, I have recently picked up work on the bacteria also. So, whenever we are talking of the antimicrobials, we talk of variety of microbes. All of them, these microbes, they are submicroscopic. That is, they cannot be seen with the naked eye, and we need at least one light microscope so that we can see them. So, in this topic of mitochondria and drug resistance, if you see when we are talking of the mitochondria, that means definitely we are not here talking about the bacteria because they are the prokaryotic pathogens. And if we talk of the bacteria and fungi, which are two most common pathogens, then bacteria, because they are prokaryotic, that's why they do not have the membrane bound organelles, and that's how they are a lot different from the humans, which are eukaryotic. Whereas in the case of the fungal pathogens, which are eukaryotic, thus they share a lot of similarity with the humans, which are their host when we are talking about the pathogenic fungi. So that gives us the problem that very few unique pathways are left that can be targeted for the discovery of the antifungal drugs. So that is one major problem that is there in the discovery of the antifungals. So if you talk of the fungal pathogens, there are different species. We already know that fungi have taken up a whole kingdom in the five kingdom classification. So different species are there. Some of them they are like Candida albicans. Then there are different species of Aspergillus. We have Cryptococcus neoformans. Then there are new species which are coming up. Like in 2009, there was one species of Candida, which was Candida auris. That has recently gained, I mean, the research on that has gained momentum because this is a multi-drug resistant pathogen. So inherently, this is resistant. So these different fungal pathogens, they cause variety of infections. They can infect our brain, they can infect our lungs, vagina, skin, and these variety of pathogens which can cause both topical and the systemic infections. That is, the infection can reach the blood, and that's how it can be disseminated to the different areas in the body. But the problem with the fungal pathogens, apart from the very limited unique pathways, is that these and the antifungals which are available, they could have toxicities issues. Then there are chances of gaining resistance against the limited antifungals which are available, along with the emergence of new pathogens that I've already told you about in the case of Candida auris. So, one problem which I already told you about, the problem does not stop here. The other challenges are there that the fungal kingdom, because it involves variety of species, that's why we see a lot of heterogeneity among the species. Some of the pathogenic fungi, they could be unicellular, like we have Candida albicans or we have Cryptococcus neoformans. Some species which are multicellular and they also cause infections, like there is Aspergillus. Apart from the unicellular or multicellular nature, some fungi, they are haploid in nature, like Candida glabrata is haploid, Candida Oris is haploid, whereas if you talk of the Candida albicans, this is a diploid pathogen. So if you're talking of the diploid fungi, that means whenever we need to work on any novel gene, we need to mutate two copies of the gene so as to render them totally ineffective in terms of their expression. Then some fungi have certain special features like Cryptococcus neoformans is known to synthesize melanin, the same melanin pigment that we have, that we call that we have during our tanning, skin tanning. So that melanin is also produced in Cryptococcus neoformans. Then it can make capsules which make them resistant to the action of the phagocytic cells. Melanin give them protection against not only against the UV rays that we know about, but also from the ROS, which is produced in the body, that is the free radicals. Then some of the fungi also pose the challenge that they do not use the standard codon usage. We know that when the central dogma we talk about, there is DNA to RNA and there is protein synthesis. So we already know about there are 64 possible combinations and how 20 amino acids they are produced. So Candida albicans, like they do not follow this standard codon usage. So whenever we are expressing their proteins in some heterologous system that is outside the Candida albicans body, like in the case of Saccharomyces cerevisiae, then we have to take care of that at least some such codon is not there, which will be giving some different amino acids than it is giving it in the case of the Candida albicans. Then you already know of the most common fungi, that is the Saccharomyces cerevisiae, which is non-pathogenic, but the other pathogens that we will be talking about. So this one difference is also there, and still, I mean, because there are a variety of species, we cannot pinpoint that only limited number of genes. I mean, these limited number of genes are causing the pathogenicity. So in the evolution, we are still not able to narrow down the genes which are responsible for causing the pathogenicity. Then as far as the infection is concerned, some of the fungi, they cause the topical infections, whereas the other, they can reach to the blood. One interesting feature of the fungi is that the single cell, like in the case of the albicans, this can form hypha under certain inducing conditions. Then there is biofilm formation, that is this cell, if it starts secreting the mucopolysaccharide, and all the other cells in the vicinity also start secreting the mucopolysaccharide, then these cells get entangled in a sheet which is known as the biofilm. And if this cell in this single form is sensitive, that is, it can be killed by the antifungals, but when it is embedded in that polysaccharide matrix, then these cells, they act as the resistant cells because sometimes the antifungus, they are not able to reach. So these are the few problems that we have when we want to design any antifungal as far as the, I mean, these problems are concerned. So it becomes really difficult. Uh, During Corona time, we all heard of the black fungus, which was caused by mucus, different mucus species. And with this, I would like to tell you that these different species that we are talking about, they are generally opportunistic pathogens. That is, they are there in the environment. They do not cause the disease. But if immune system of the person becomes weak, like in the AIDS patient or person who is undergoing some hormonal therapy or transplantation patients which have been put under steroidal therapy, then because their immune system is not able to fight the infections, then these infections basically take over and it can become life threatening also. We already know of the resistance emergence that we talk in bacteria, that if we do not complete our course, then the resistant pathogens, they will not be killed, and later on they will outgrow and the resistance will emerge. So like in the case of the bacteria, there are different mechanisms of resistance in the fungi also. And if you look at this slide, this is showing you the different mechanisms which are involved in the resistance in the fungal pathogens. The first one, if you see, this is basically showing you the different proteins which are there in the plasma membrane, and during the resistance cases, their number in the membrane may increase, and thus if any drugs which is coming inside, it will be expelled outside, and thus enough cytotoxic concentration inside the cell is not there to kill it. That is one of the chief mechanisms. Apart from that, sometimes the target of the drug, which is there inside the cell, that gets mutated and the drug is not able to bind onto that. So even if you have the enough quantity of the drug inside the cell, but because it's not able to recognize the target, thus the cell will become resistant to the drug. Sometimes these drugs, when they enter inside the cell, they can be rendered ineffective because of the modifications that can happen on them, or they can be sequestered in some organelles, and thus they are not able to perform their functions. So these some of them, they are the chief main mechanisms that basically contribute to resistance. If you see this slide, this is showing you the different antifungals that have been discovered on a timeline scale. And if you see, there are very limited number of the antifungals which came to the market and which were FDA approved. In this 70-year lifespan, the last one, they are from the echinocandins group. So all these antifungals, they basically belong to four different categories. The first one bind onto the ergosterol pathway. Like we have cholesterol, humans do have cholesterol, then the fungal cells do have ergosterol. So since ergosterol is structurally different, so is the pathway which is involved in their biosynthesis. So some of the drugs, they target on this pathway, like there are azoles, including fluconazole, ketoconazole. Then there are allylamines like terbinafine, which bind to the very first enzyme in the ergosterol biosynthesis pathway. Then second one, I would like to tell you about the membrane, cell wall membrane. Here, the polyenes drug, they also bind onto the ergosterol, but not the biosynthesis, but the ergosterol which is present in the membrane, that is basically bound. And after binding of this ergosterol, the membrane fluidity changes, and that basically result in the more drug being taken up inside and results in the killing of the cell. Then there are nucleic acid synthesis inhibitors like 5-flucytosine. And last but not the least in this category is the latest drugs which belong to the echinocandins category, and they bind to the cell wall synthesis. And they include caspofungin, then micafungin, anidulafungin. So all these are different variants of the echinocandin drugs. They work on the beta glucan synthesis enzyme, which is involved in the chitin, which is present in the cell wall. So limited drugs are there, which came after a long span of time, and resistance has already been reported against all these antifungals. So that is a matter of concern, that we need to look for the new drugs for which we require new novel targets. So in the year 2022, this after the corona, WHO basically came up with a list of the fungal pathogens which can be a threat to humanity. This list include 19 priority pathogens which should be taken up for the antifungal discovery. And in this, four fungi which have topped in this list. That includes Candida albicans. Then there is Candida auris, which I already told you about, which was discovered in 2009 in Japan. Then there are different species of Aspergillus, and last but not the least is the Cryptococcus neoformans. In our lab, we are working on these three fungi, and this piece of work that I'll be discussing will be basically include the work that we did on Cryptococcus neoformans. So during my early career, when I started with my research, I basically worked on the fungi and how the lipid composition of the fungi basically affects the drug resistance pattern. So in that, we got to know that since the plasma membrane comprises of different lipids, like we have sterols, we have sphingolipids, we have phospholipids, so they play an important role, because when we change their composition, then this changes the fluidity of the membrane, and it can affect the import as well as the efflux of the drug. Then later on, when I joined here in MD University in Rohtak, and I was setting up my own lab, then the first piece of work that I started with was the phospholipids. And you already know that phospholipids, they are a major component in the plasma membrane. There are different phospholipids, like there are phosphatidylserine, there is phosphatidylcholine, ethanolamine, and these different variants, they are present asymmetrically in the two leaflets of the plasma membrane. If you know of, it is very interesting to hear, because these different phospholipids, because they are asymmetrically distributed, if their pattern changes, especially phosphatidylserine, which is, they are more in the inside leaflet, which is facing the cytoplasm. If it comes to the outside, then it can result in apoptosis and killing up of the cell. I was not interested in apoptosis. I was just, telling you about the phospholipids and its importance. So that piece of work basically led us to identification of the role of mitochondria in drug resistance. Phosphate biosynthesis happen in the vesicles inside the cytoplasm, as well as in the mitochondria. One of the enzyme that is coded by the PSD1 gene, that is present in the mitochondria, and that's how this is important in the synthesis of two different phospholipids, that is phosphatidylserine and choline. So mitochondria, you know of, it is a semi-autonomous organelle. It has its own genome, but that doesn't mean that it is working totally independent of the nucleus. There is a signaling that happens from the mitochondria to nucleus, that is known as the retrograde signaling. Generally, we know that nucleus is the chief organelle that controls all other, functions inside the cell. Here, in case of the retrograde signaling, the signals comes from the mitochondria that reaches to the nucleus, and then nuclear expression basically controls the different downstream pathway. So when we were studying about more about this role of mitochondria in the phospholipid synthesis, then we identified this complex, which is known as the IRMIS complex, and it stands for the endoplasmic reticulum mitochondria encounter structures. This basically is a multi-protein complex, and it joins the ER with the mitochondria, which was very interesting at that time when we realized, because in our books earlier, we always used to read the organelles as the independent sites where they are performing their unique functions. And unlike the prokaryotic cells, there is no mixing up of the functions. So why do, these connections between the organelles, they exist? So that was pretty interesting as far as the resistance is concerned. So ER and mitochondria, they make variety of structures. It's not the IRMIS, but there are other proteins also that connect. But this protein complex is unique to the fungal kingdom. That means it does not occur in humans. So if you want to target, any protein or any gene which is unique to the fungi, that can be studied for the development of the antifungals. So this protein complex comprises of 4 subunits, which are triple M1, MDM10, MDM12, and MDM34. So if you see over here, this triple M1 is present on the ER membrane, whereas MDM10 and 34, they are present on the outer mitochondrial membrane. This MDM12 is a totally cytosolic protein and is tethering these two triple M1 and MDM34 protein. So these information, they were obtained from the earlier work, including the bioinformatic work which was done, how these proteins, they have certain domains which are known as the lipid domains, and they might be involved in the transfer of lipids between the organelles. So because of the uniqueness, we were interested in reading more about this complex, and looking at the different reports which were available, we studied that these 4 genes have already been studied in different fungi, and deletion of all these genes result in the changes in the mitochondrial morphology. Since we earlier found that in our studies that mitochondria could be important in regulating the drug resistance, so the unique nature and the changes in the mitochondrial morphology made us interested to pursue this further. At that time when we decided this, there were 2 or 3 reports. One was in Saccharomyces cerevisiae, where they showed the change in the morphology, and apart from that, there was another report in Candida albicans, where they mutated this and they found that they made the cell sensitive to one or two drugs. So it was a very preliminary study. And for our work, we chose Cryptococcus neoformans because this fungus is known to cause certain outbreaks in some part of the world, including in the USA. And the related species, the Cryptococcus gatti, caused one outbreak in Canada in the Vancouver Island during 1999 and 2007. So I never worked on this fungus earlier, but it was a challenge to start up with something new. So during the studies on and the initial studies on this fungus, we found that apart from the challenges on working on a new fungus, one chief problem was that this fungus has certain unique features from the candida on which I was earlier working on. One was it was a haploid fungus, whereas Candida albicans is diploid. So that worked in our favor. That means we had only one copy of the gene to delete with. Then we were lucky that the genome was already sequenced, it was available, but it had a capsule, and along with that, it used to make melanin sheet. So these two were new things that we were dealing with for the first time. Capsule gave it a problem that when we want to delete a gene and we have to introduce some DNA from outside, then it is difficult for the cells to take the DNA of interest. So if you look at the structure of the membrane and the outside the plasma membrane, if you see there is deposition of these melanin granules, which is shown in black color. Then outside is the cell wall, and further to that is the capsule. Uh, the next picture is showing you how the colony looks like after the melanin synthesis, and that's how we see the Cryptococcus colonies when we do not have any inducer for melanin synthesis. So till now, we just knew a little bit about the different contact sites which happen between the ER and mitochondria. But later on, we realized that mitochondria is in contact with different other organelles, which are known as the mitochondria contact sites, which include the peroxisome, lysosomes, lipid droplets, melanosomes, and Golgi apparatus. So all these, we are still, moving on to them, how they can affect the change of the metabolites between the mitochondria and the ER. But to start with, we restricted ourselves to the Ilmis complex, and these 4 genes we decided to go further with. So we procured the strains and the 4 genes, the sequence was already available. We procured the sequence, and then we decided to delete them by making the deletion cassettes. In fungi, one of the ways to delete a gene is homologous recombination. So for that, what we need to do that is that the intact ORF copy need to be replaced with the DNA, which is having the flanking regions which are same as the targeted gene, but the ORF is replaced with the selection marker. That is the drug on which we can select the colonies after transformation. So in this case, what we did is like instead of cloning, we use the triple joint PCR for these 4 genes. We PCR amplified the 5' and the 3' region of the genes with the primers 1F and 2R, and then 5F and 6R. So these 2 flanking regions, then they were ultimately joined with the selection marker that gives the resistance to geneticin. All these 3 fragments, they would have the overlapping sequences. So they are mixed together, and in a common PCR, they are all joined. This results in the bigger fragment. And with these 2 primers, the terminal primers 1F and 6R, we amplify this whole cassette and we replace the intact copy of the gene. Thus, the ORF is basically not there to make the mRNA and the protein. So like this, these 4 copies of these deletion cassettes, they were introduced. And as I told you that because of the capsule, we had a lot of problem because the transformation efficiency was pretty less. But ultimately, we collected few of the colonies and we confirmed them that the transformants which we obtained, now they have become resistant because of the gain of function for this resistance marker. These colonies, they were confirmed with the southern analysis and with the RT-PCR, and it showed that there is no expression of even a little bit of the transcript of the four genes that we wanted to study further. In cell biology, if you want to study any gene, generally there are two ways. Either you delete the gene or you overexpress the gene. Since we decided the first way, that is to delete the gene and to check their role in affecting the drug resistance, it was important to see if the deletion of these genes is vital to the activity, to the survival of the cells or not, so that it does not hamper the analysis with our results. Luckily, the four genes that we deleted, that were not affecting the growth of any of the mutants. And this was done both at 30 degrees Celsius and 37 degrees Celsius, which is the physiological temperature, which was interesting because in the case of some other fungi, like in Saccharomyces cerevisiae and Candida albicans, this triple M1, which is present on the ER membrane, that generally is not tolerated by the cell, and its deletion results in the growth defects. Thus, they have to make some conditional mutants, and by the addition of the repressor, they silence the gene and then they check the phenotype of that particular mutant. So after confirming that the deletion of the genes is not causing any problem, we quickly moved on to see the deletion of these genes on the drug sensitivity. We tried different drugs, and the interesting results were seen first in the case of the fluconazole, which is the most common antifungal pathogen. It belongs to the azole category and bind onto the ergosterol pathway. The two mutants, that is triple M1 and MDM34, gave the sensitivity. As you see, in case of wild type, 2 microgram of the drug is able to kill the cell, whereas in the case of these two, we require very less drug than the wild type. That means these two mutants are becoming sensitive, which was interesting. The next antifungals that were showing quite interesting results were the echinocandins. The caspofungin, micafungin, and annurilafungin, they all belong to the category of the echinocandins and work on the cell wall biosynthesis. All these three drugs, they were rendering these four mutants sensitive, whereas no such change was seen in the case of the amphotericin B, which also work on the ergosterol synthesis, but it's a polyene. Cyclohexamide and flucytosine, they were also causing the sensitivity, but it was not that promising. Only slight change was there. So with this, we summarize that it is the echinocandins which are causing the most interesting results as far as the sensitivity is concerned. So with this, the next piece of experiment that we did was on the mitochondria. Since the earlier two reports that were already published before starting our work, they showed that there is change in the morphology of the mitochondria of these mutants. We wanted to confirm if this is the case in the case of the fungi C. neoformans as well. And here in this case, if you see, there is a staining of the mitochondria, which is done with the red tracker dye. This is specific for mitochondria. It's a MitoTracker CXM ROS dye. And when we incubate the cells with this dye goes inside the mitochondria. And if you see in the case of the wild type cells, we see these tubulated mitochondria, whereas in the case of the mutants, we do not see this tubular morphology, rather we see patchy fluorescence, and that shows the fragmented staining pattern. So this was quite similar to the earlier reports that we saw. So this indeed proved that there is change in the morphology of the mitochondria when this illness complex integrity is compromised. Since we were sure of the morphology now, we wanted to check if the functioning of the mitochondria is also affected. So we already know that fungi, they are having fermentation as well as the respiration which is happening. So when we use the glucose, so cell has the option of basically, this is fermentable glucose. So in this case, when we check the growth of these wild type cells and the mutants, then the cells were growing happily on the glucose, which was fermentable carbon, but we use the non-fermentable carbon, then the cells were not able to use them. The mutants, they were not able to use them, but the wild type, they were living happily. So with this, we summarize that the IRMIS mutations, the IRMIS deletion results in the change in the morphology of the mitochondria, and it causes the respiration defects in the cells. Since mitochondria is the site where a lot of ROS is produced because of the electron transport chain, so we wanted to see if there is any change in the ROS levels. So for this, the first experiment that we did was to check the sensitivity of these mutants to the hydrogen peroxide, which causes ROS stress. And we saw that an interesting contradictory result is that the triple M1 and MDM10 mutants, the first two mutants, they are showing better results, that is better growth than the wild type, whereas the MDM12 and MDM34, they were growing less than the wild type. Then to make sure that the change in the growth is because of the hydrogen peroxide, we used ascorbic acid, which is able to reduce the ROS levels, and we saw that addition of ascorbic acid indeed resulted in the regrowth. So this indeed confirmed the phenotype that we got over here. Apart from that, we also checked the ROS levels with the fluorescent dyes. There is a dye that is known as dichlorohydrofluorescein diacetate. This is commercially available, and this basically shows more fluorescence if more ROS is there. And we saw that in the case of the triple M1 and MDM2 and MDM10, there is less fluorescence here, suggesting the ROS levels basically decrease. And that is the reason that more growth is seen here, irrespective of the fact that H2O2 stress is there. Whereas in the case of the rest of the two mutants, they had more fluorescence than the wild type. These results were also checked quantitatively by taking their fluorescence intensity in the fluorimeter. And if you see the triple M1 and MDM10 mutant that we had, they had the reduced fluorescence intensity, showing ROS levels reduction in these two mutants. So in our earlier results, we already observed that the cells became sensitive to the echinocandins drug, but the sensitivity with the other antifungals that were not pretty, interesting. So we came to this conclusion that we should definitely go for the biofilm synthesis. And as I already told you that the single cells, they could be sensitive, but when they start making biofilms, sometimes they become resistant to the drugs. And that is one of the problem when we are treating with the fungal pathogens, because in the body, if they come across any catheters or any IV devices, then these fungal pathogens, they can start making these biofilms, and thus the patient, even if they are kept on the antifungal therapy, they are still, have this fungal infections for many years. Interestingly, we saw that when we compared the mutants with the wild type, there was no change in the biofilm formation in the absence of any drug. Whereas, just like the planktonic cells, the single cells, in the case of the biofilms, the mutants, they were making less biofilms. That means they were sensitive to these antifungals. So these biofilms, they were dissolved with ethanol and they were collected, and the data was taken by the spectrophotometric readings, and indeed we confirmed that there is significant change in the biofilm inhibition in the presence of these antifungals. So with this, we can say that these mutants, they are causing a change in the drug sensitivity to antifungals, and they are causing a change in the mitochondrial morphology and causing the respiration defects. Since I already told you that C. neoformans is unique in terms of making the capsule and melanin, and these two happen to be virulence parameters for C. neoformans. So we decided to see its effect on the capsule and melanin, and we saw that in the mutants, if you see in the triple M1 and in the case of the MDM10 mutants, the capsule thickness basically reduces, whereas the change was not much significant in the case of MDM12 and MDM34. Similarly, if you talk of the melanin pigment, which gives the protection to the fungal from different kind of stress, triple M1 and MDM2, they are not making any melanin, whereas in the case of MDM12 and 34, not much difference was seen. So here you are seeing the cells. These cells, when they are collected in the 96 well plate, so a single well is basically is taken over here and is shown so as to show the difference in the pigmentation. These data, they was quantitated and then this is shown here that the pigmentation basically is changed in these mutants. So with this, we were sure that mitochondria is definitely getting affected. But in the earlier papers that we talked about on the limited species which were available, they never talked about the partner organelle, that is the ER. So what is happening to the ER? We already know that ER is the site where a lot of protein synthesis happen. And these protein also include the enzymes which are important for the fungal pathogenesis. The enzymes including phospholipase, urease and catalase, they are important. Like phospholipase basically causes the breakdown of the lipid molecules, different lipid molecules, and that's how this results in the, or this helps in the spread of the infection to the deep-seated areas. So this will break the tissues of the body and then the fungal can spread to different parts. So the only method we had was to look for the activity of these enzymes, if these enzymes they are produced or their activity is affected or not. So we selected these
और
enzymes.
मैडम कितने स्लाइड्स हैं?
I think 2, 3 more.
ठीक है मैडम, कर दीजिए.
So these enzymes, when we checked on different plates, like this phospholipase is checked on the egg yolk agar, urease is checked on the Christensen urea agar, and catalase is checked with the presence of the hydrogen peroxide. So we found that in the case of triple M1 and MDM10, the activity is reduced. So that means it is not only causing a stress to the mitochondria, but indeed ER is also affected. Similarly, if you talk of the hemolytic activity of the fungi, that is also reduced. And these results, they are quantified here. And now we were sure about the effect of the IRMIS on the ER. Then apart from that, another test we did that was on hydrophobicity. And hydrophobicity of these cells is important because that affect the adhesion of the cells. When these cells, they have to cause the deep-seated infections, firstly they have to adhere to the host and then they basically start spreading. So this test is done with the N-hexadecane, which is mixed with the with water, distilled water or buffer, and then we see the separation of the cells in these two phases. That is the aquatic, that is the aqueous phase or the organic phase. So we saw that comparative to the wild type cells, the mutants, they have some changes in the hydrophobicity and there are more cells in the aqueous phase, in the lower phase. And that was interesting because maybe that is the reason that we see changes in the virulence of these mutants. So till now, what we concluded is that there is change in the mitochondrial morphology and it is causing the respiration defects. Along with that, the cells become sensitive to the echinocandins drugs in both planktonic and biofilms. There were change in the ROS levels and there was reduced melanin and capsule thickness in the senior-formant cells. The enzymatic activities that we checked, they were also found to be affected and there was reduced hemolytic activity and the adhesion of the cells. So they were all in vitro results and they were interesting and they were all suggesting that the cells, the mutants, they might be now less powerful in causing the infection now, and that could be validated only with the in vivo results. And since we were just starting with this kind of work, we used the C. elegans, synorbitated elegans infection model. And these C. elegans, basically they are nematodes and they are used as a model system for different fungal pathogens. In this case, when we have the candida or cryptococcus going inside, this causes the rupturing of the organs. So it is able to mimic almost the innate immune system of the humans. So these nematodes, they are taken and they are fed with these fungi, and we found that when we use these mutants, then if you look into the this survival curve, then this red line is showing you the wild type cells. That means they are getting killed. This is killing in the nematode after 5 or 6 days, whereas these four mutants, they are surviving in those nematodes for a longer time than these cells, and they are not basically causing the killing of this particular nematode. Along with this survival curve, we also looked into the organs. So the black, these triangles and the white triangles that you are seeing, these are the different organs. These are the pharynx and the intestinal region. And we found them that they become totally distorted than the typical shape that we see in the wild type. And that confirmed that the sensitivity basically is seen in the in vitro assays that affect the virulence of these basically pathogen also. So this study that has been published in different papers, and we are still continuing with this work, and hopefully it will be in the domain soon. So that work that we worked on the cell wall of the fungi, including we took the electron microscopic picture. That results I'm not showing here, and with this. I would like to summarize whatever I did, and these papers are already in the public domain. You can read them. The protocols that have been standardized during this study, that we compiled all of them for the availability of the students, and they have also been published just 1 month back. So this is my lab. If you see my students, they are here. And I would like to acknowledge to the funding agencies. We received funding from DBT, UGC, then from the university, and also from the SERB, that is from the DST. The work that I presented here is basically
Yes, yes, बहुत.
PhD work of these two students, and both of them, they are Deepika. And last but not the least is all the scientists who basically made the not only the strains available, but also they opened their labs for the training and for doing the experiments that were badly needed to complete these studies. So
Thank you,
with this,
madam.
I would like to thank all of you for your
Yes,
patience and your
yes,
time, and I can now
yes, yes.